all. So today I just want to briefly kind of go over how I deal with phasing out haplotypes on nanopore sequences using the program Ugene. So right here we have on Micromap our page with all of our data files that we need. So I download the assembly file here, and then I'm just going to copy that original sequence. So open up that FASTQ, join the sequences into alignment, and open in multiple alignment viewer. Now the first thing you want to do, and you always want to do this, is just save the alignment as, and then align it. I usually use MAFT as my initial alignment. It's quick and it does the trick. And then while that's thinking, I'm going to also paste that original consensus sequence into Ugene. I use it as like a reference sequence. Export as alignment, just to give me a quick visual to see when I might have a sequence that is in need of editing. And you'll see why in a second. So Control F to search for a conserved sequence, in this case agat-kata. That'll tell me which group of sequences is the forward read and which is the reverse. So this bottom group is the forward. So I'm just going to reverse complement the one I need to reverse complement, align again, and this should get me basically where I'm looking to be. Copy consensus and paste that over, and then align that. Now, it is possible to copy, paste that consensus sequence that ng species ID spits out and use it as a reference sequence. So you can set it as the reference sequence. I don't find that super useful because the way I'm using it, I'm basically using it to find spots where there might be an ambiguity, right? Just save myself a little bit of time. I am often processing a lot of these at once, and I don't want to have to sit here and like scroll to find, okay, see, it just takes so long. There's an ambiguous position here, right? I don't want to have to scroll and find them manually. I can just do it this way. So for my sanity, I'm going to rename these F2, just so I can keep track of what I'm looking at. And then we're going to trim this here and trim this here. Now, see at the bottom here, there's a bunch of these spikes where there is a bit of disagreement. So at this point, I usually just kind of go through and start taking a look at what's going on. You may ask yourself, how do you get to the setting? These are the settings I'm using. I'm using consensus type simple extended of the threshold of 70%, which will more or less kind of get you this. And that's, it's erring on the side of caution. Like, this is TCA. Like, I'm not concerned about that being something different, or TAT. It's only about 10% difference. So we'll just change that back. Here, we might actually have a real ambiguity. So we're going to copy over just a group of nucleotides that'll help us search for the right spot. And we see, yeah, there's a pretty good chance this is actually a real difference. So the thing I'm going to do next is align with muscle. Just to show you what I did again, I hit do not rearrange sequences. So uncheck that box. And that means that when it goes and aligns it, it'll rearrange the sequences for you. No, and sometimes it ends up with a situation where there's some extra noise at the top, so not everything aligned perfectly properly, like there's some noisy sequences in here. And that's okay, because Nanopore works off of a consensus of multiple sequences, so that, if some of them aren't quite right, it isn't going to throw the entire thing off. Heard? Okay. And honestly, I would not be surprised if I blasted just this little chunk here, if it might be something slightly different. It could be a contaminant. I'm gonna realign these. That chunk is looking awfully funky. Okay, I'm actually gonna do something first. We're going to export that. And we're just gonna we are going to just do a quick and dirty blast on that to see if it is just a contaminant, then we should get rid of those sequences. So, because sometimes you do get some like yeast or other fungal contaminant sequences kind of mixed in here. And I don't necessarily want to be using that. Okay, so this little chunk is Lacaria. This is not my target specimen. So this is either a little bit of bioinformatic contamination or a little bit of physical contamination. So this particular chunk right here is no bueno. So we're just gonna take those out, leave those out, delete them. We don't want them. Now we have a little bit of a cleaner situation happening. Don't do that. So let's try this again. Alrighty. So we got my Y's, my R's. Yeah. So that looks a lot cleaner. It's still not perfect, but I'm not concerned over much with a couple aberrant reads because it should balance out, right? We're working off averages here. We're not working off of, you know, every read is going to be perfect. This is why Nanopore works, because it works off of averages. So we're gonna, take everything below this spot, split this off into a new alignment. And this will be my haplotype B, this will be my haplotype A. So next thing I'm going to do is copy consensus, rename it, paste and rename it, copy consensus, paste F2, hap B. And then we're going to align again. Okay. Trim the ends, I don't want this. Alright. So now, what I usually end up doing is kind of going through everything manually just to see what's going on. Because you do run into occasional wonky situations where there's an alignment issue and it didn't quite detect what was going on. Like right here, I'm going to guess the initial alignment was just kind of wonky. Normally it's not an issue, but when you get these polymorphic sequences, sometimes you do get some strange alignments with the computer algorithm. Now I know this G, this R, and this Y are definitely there because I already checked them. So yeah, A, G, A, really solid. A, G, A, again, super solid. That Lacaria may have screwed up my initial sequence, so we're just going to get rid of that. Now this M looks like it might be a real, so we're going to check him. So we got an A, a pretty good A at that position. Same thing on the other one, and we have a not finding that. 1 sec. There we go. And we have a really strong C at that position. So that M is definitely real. T, 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 3 T's, 2 T's. So yeah. So this means that this haplotype B has there's an indel at this position between the two haplotypes. In cases like this, I have been erring on the side of adding the extra information into my consensus, just to my consensus is going to have the most information basically. So there's no IUPAC ambiguity code to say, oh, one haplotype has a gap and one haplotype has a T. So we just kind of have to do the best we can. Again, right here, that's probably from some alignment wonkiness. Yeah, those are definitely, that should be a C. Two, three, four, five, six, seven. We'll just get rid of that and make that an A. I'm guessing, like I said, I'm guessing with that initial alignment, that Lacaria chunk that was in there was causing some issues. But yeah, we're just going to kind of keep going through this and checking them to see what is, going on with the alignment. We're going to find the actual ambiguous positions. That's a T. And it's a C, so it's definitely a Y. And basically my goal is I just want to try to have the most accurate data that I can. It's not going to be perfect, but we can do our best. And that's okay. So that's definitely an A. That's an A. That's a G. So that's an R. Didn't realize this one would be quite so time consuming all. Sorry about that. A. It's GA, so that's definitely an R. That A certainly doesn't belong there. GA. Definitely GA. Not as strongly expressed. And I do find, so Nanopore occasionally has trouble reading these homopolymer repeats, like these repeating regions. And so you'll end up with situations where, there's a shift of like one frame here and one frame here and one frame here, right? And I could, if I want to, start going through and like adjusting that alignment manually. But in this case, I'm going to say I'm fairly confident that it should just be GA right here and not a different ambiguous code. And it is definitely GA on this side. So as far as I'm concerned, there's not really much ambiguity there. Okay, so after cleanup, we ended up with 1, 2, 3, 4, 5, 6 ambiguous positions and then 1 indel, right? So what I would then do, if you want to continue exporting this to Micromap and INAT, is, so kind of go back here. I'm going to get rid of that. That was my dirty list. So what I usually do is the first thing I'll do is copy my consensus, and Control C is going to be Control C copies a single selected line, right? So pop my edited sequence in there. While it thinks. Then we're going to also make separate blasts for hap A and hap B here. And then I probably should open up my INAT report association. So to create those new mycoblasts, open up mycoblast search. This is the entellome I'm looking at, by the way. All right, so the next thing I usually name them like this. So I'll just create a blast that says hap A and a blast that says hap B. And then just copy, Control C, the relevant sequences. Oh, ooh, I was a little quick on that. Make sure you delete any gaps. All right. And then you'll get a blast that has the individual haplotype blasts. Then I'm also going to rerun the blast for the one that I edited. So I have my edited consensus sequence. I have haplotype A split out, and then I have haplotype B split out. And they're definitely, matching slightly different things, but once I get this entire group cleaned up, I'm going to take a different look at it. There's a lot of sequences I'm cleaning up here. And then the last thing I'm generally go do is, let's see, how many did I say? 1, 2, 6. Because you can't really put extra haplotypes on Micromap, but you can definitely put it on iNaturalist. So I have these fields we've kind of created to make this a little more organized. So haplotype A, DNA barcode ITS, haplotype A, mycoblast search, haplotype B, DNA barcode ITS, haplotype B, mycoblast search. And then also we want to add the edited original sequence here. And we want to update the blast to the new blast for the edited sequence. And there you have it. Haplotypes all phased out. The information's there. So if you need to look at it later, or someone else needs to look at it and see what's going on, they kind of have the full information instead of just, the initial sequence that was spit out that didn't account for the ambiguities. Hopefully that's helpful. Have a good day.